and how that's good. We know how ABCA1 can be degraded through the escort lysosome pathway, and that's bad. And we know that ubiquitination normally regulates the sorting of proteins into the escort pathway. Let's put it all together and let's say that what pathway marks ABCA1 for endosomal sorting is still unknown. So that's the way authors have to say, this is important, this is important, this is important. Now we put it all together and there is something that is clearly important that we don't know yet and that we'll address with our work. And bless you. And the last bit is that Listerine is an E3 ubiquitin ligase that is regulated in cholesterol-stimulated macrophages. So essentially with that, the authors formulate the driving hypothesis behind this paper, and that is, could Listerine be the one E3 ubiquitin ligase that regulates ABCA1? And it's in the paper's title, the E3 ubiquitin ligase. We bring it up again as a concept here. Very briefly, in the process of polyubiquitinylation, there is typically 3 enzymes involved that are called E1, E2, and E3. E1 is the one that prepares ubiquitin, E2 is the one that activates it, and E3 is the one that normally matters the most because it's the one that ligates that activated ubiquitin molecule to the target protein in question. So there is a handful of E1s and E2s, but there is many, many, many different E3s that differ among them by the type of target proteins they work with. So Listerine could be, these authors propose, could be the E3 ubiquitin ligase that might be attaching ubiquitin molecules to ABCA1 to potentially regulate its sorting into the escort lysosome pathway. Questions or comments so far? Now we're good? All right, cool. So let's dive into data now. The authors cite a bunch of previous studies that they had conducted linking the expression of Listerine to progression of atherosclerosis, right? And one piece of data that they reproduced in this paper is this. They take ApoE mutant and LDL mutant mice. These are mice that by definition are going to have a much harder time dealing with cholesterol and other lipids because they don't have ApoE and LDL, both proteins that normally sequester bad lipids from cells and blood and interstitial fluids. So these are mice that are predisposed to hyperlipidemia and things that are related to hyperlipidemia like atherosclerosis and presumably Alzheimer's disease. And they take these mice and put them on a Western diet. Quotation marks there are to indicate that that's how they refer to this diet in the paper itself. It's a highly caloric, highly a high-fat content diet. And they know that by doing that, taking these mice that are predisposed to hyperlipidemia-driven conditions like atherosclerosis on a Western diet, they will more likely develop atherosclerosis over time. And what they do is they isolate macrophages from these mice at various time points, 0 weeks, the beginning of the assay, 8, 16, 26 weeks, and they measure the expression of Listerine from these macrophages, and as you can see, there is this progressive, significant accumulation of Listerine in these cells. So that is from mice that are predisposed to hyperlipidemia. In humans, if you take macrophages from control individuals without a condition or individuals with atherosclerotic cardiovascular disease, you will see that there is a significantly higher levels of Listerine mRNA in human atherosclerotic plaques, and the same goes if they measure Listerine in blood, right? Presumably blood cells, obviously. So all of these are, if you will, epidemiological observations of a correlation, if you will, between Listerine levels and progression of atherosclerotic disease. So they wanted to take a little bit of a more of a proactive approach. They took peripheral macrophages and they exposed those macrophages to these oxidated LDL molecules that sort of mimic a very hyperlipidemic environment for these macrophages. So these macrophages are sensing this very high lipidemia, and what they observe in vitro is that Listerine expression starts going up within a day, essentially. Within 24 hours, there is this very drastic increase in Listerine expression. And they see that at the protein level, so these are Western blots, and they do the densitometry, of course. Let me send that for the video. But they also see that at the level of mRNA levels by quantitative PCR, right? Or RT-qPCR. All right, so as macrophages live in hyperlipidemic environments, in vivo environments, or as they are exposed to very high fat content in a Petri dish, in both cases they express higher levels of Listerine. And in the text, the authors write, taken together, these data suggest that Listerine is involved in the development of atherosclerosis and plays a potential role in this disease. And what I wanted to do now is invite everybody to take a few seconds and let these statements sink in. So taken together, these data suggest that Listerine is involved in the development of atherosclerosis and plays a potential role in this disease. My question to you guys, and I hope that we can have some discussion, is do you agree with this statement? And if not, how would you rephrase it? Association, or what's the one that we throw around with causation normally in that famous saying? Correlation, right? You've heard this before, right? Correlation is not causation. And that's exactly right. Here, what they've shown us so far is a correlation, not causation. So how do we rephrase it to indicate that? You said, well, I'd say there is a correlation, not causation. This sounds a little too much like there is.
Listerine has correlation with the development of atherosclerosis, but necessarily not causation.
Okay, all right. Any other ways of rephrasing, anyone? You guys agree with that? So that's certainly a possibility. You're very explicit and you say we observe this correlation, which of course doesn't mean causation. You know, one could be very explicit. This language here, the reason I wanted to quote the language exactly is because this language that we see here in this paper is pretty common in papers. If you were to say to the authors, if we had them in front of us and we say, hey, you're implying causation, you're saying that, I don't remember exactly your words, but you said, you're making it sound like this thing causes atherosclerosis, but you're not showing any evidence for that. So if we were to say to them that, they would probably say, whoa, no, we never said that these data demonstrate causation. We're just simply saying that the data suggest, right? And then we say that there is a potential role, right? So the reason I want to bring it up is because this language, even though it might come across like a little, what's the word that I'm looking for? That they're playing with words, right? It's fairly common. It's fairly common. And it comes from the fact that it's true, they're not showing causation, but it is also true that one of the first conditions for establishing some sort of causative role is to show precedence, right? So if I'm, at the end of the day, I'm going to make the claim that increased Listerine expression drives atherosclerosis progression, then at the very least, I should show that they're correlated, right? Either in expression or in activity. And so by showing that correlation, they're sort of checking that first box for things that you would expect of causal roles in a process, right? So these interplay between I show you a correlation and then I say that this data suggests that there is a causal role is more common than not, right? So now as you guys read more and more papers, you may bump into this rhythm of presenting the data and linking the logics towards the next experiment and think, hey, yeah, I remember that. We saw that in class. Okay? So, like you said, this is correlation, not causation. How would you address the question? Is it cause or correlation? What can you do? If you'd like to test the causality of blistering in atherosclerosis, what can you do experimentally? Gabi?
You have it there, but loss of function and gain of function experiments so that you take out Lister and see what happens.
All right, so let's go with this idea that Listerin is driving atherosclerosis. If you take out Listerin, what would you expect to see? Decrease, right? And if you put more atherosclerosis, right? So ultimately, that's what you would like to see. In this case, they didn't go all the way yet. In this figure, which I think is figure 2 of the paper, they're going for something a little more immediate, and that is their thinking, the whole thinking is that Listerin may be affecting how macrophages deal with cholesterol, right? So we're not going to go as far as to talk about atherosclerotic plaques and heart disease and heart attacks, but at the very least, let's take a look at how macrophages deal with cholesterol depending on whether they have less or more Listerin. So to take out Listerin, like you said, the approach they take is the same approach that we saw last week. They use the Krylog system. In this case, the gene that was floxed is Listerin. So that's why you see up here they have mice that are Listerin FL, meaning floxed Listerin, crossed to Liz-Cre mice. They are not very explicit in explaining this, but we need to assume that the Liz promoter obviously drives expression of Cre in macrophages, at least. Whether or not this promoter is known to drive expression of Cre in other tissues, they don't tell us. Right? If it did, then you would expect that they run some sort of control or trick like we saw last week with the LRP1 paper. But in this case, what they're showing us is that when they take mice where the Listerin gene is floxed and they excise it out using Liz-Cre, there is an accumulation of lipids inside of macrophages. So the image that they show us here is that of macrophages, of white blood cells in culture. And on top, there is the control, meaning these cells have not been incubated with high levels of lipids. At the bottom, there is an incubation of these cells with very high fat content. On the left, there is the Listerin flox mice without a source of Cre expression. So the flox Listerin gene is still there, meaning these mice, for all intents and purposes, are like a wild type. And then on the right, we have the Listerin flox plus Liz-Cre, meaning that Listerin has been cut out from the genome in macrophages, at least. Right? And the punchline of this experiment is that when macrophages don't have any Listerin in them, they will accumulate more lipids in them than macrophages that do have a functional Listerin. Now, if you compare the control mice in a control medium with, I said mice, I meant macrophages. So the control macrophages with no fats in the media and control macrophages with fats in the media, you do see an accumulation of fats in those control macrophages. And that's totally fine because that's what macrophages will do. They will take up some of that fat or those lipids in the culture media. But the observation here is that when those macrophages don't have Listerine, they accumulate a lot more of those lipids present in the culture medium. The bar plot to the right is simply quantification of what they saw through several images, and that there is that significant accumulation of lipids within the cells. It says here in the y-axis oil red staining. So oil red is a staining procedure that is very common in experimental biology. If you want to see how much the lipid content within cells is, or within tissues, you can expose your samples to this oil red. And what it is a molecule that will bind to lipids in general and fluoresces in red, or in this case, it's actually not even fluorescing, it's just showing that red color, and you can quantify that through image analysis, and that's what they show us in the graph. So according to that observation, Listerine seems to be inhibiting hyperlipidemia, meaning it's inhibiting the accumulation of lipids within cells. Then the counterpart experiment is a Listerine gain of function. So for that, what they do is they treat these cells with, in this case, these were mice that were treated with an adenovirus with sequence coding for wild-type Listerine, or for sequence coding for a mutant Listerine that is missing that ability to attach ubiquitin molecules to its targets, right? So it lost its catalytic function. And what they see, and it's a little harder, I recognize that it's a little harder to see through the images, but here we have the quantification that comes from those images. What they see is that when those cells were exposed to simply an expression vector with nothing in it, meaning a transfection, or in this case an infection control, there is X amount of oil red staining, so some baseline amount of lipids in those cells. If they express high levels of Listerine protein, that accumulation of lipids drops significantly. But if they express a Listerine that cannot attach ubiquitin molecules to its targets, it's almost like they introduce nothing, right? So it's, for all intents and purposes, there is no change based to control. What that says, and this is a theme that repeats throughout the paper, what that is saying is, it's not just putting Listerine into cells. That Listerine needs to be functional. That Listerine needs to have the ability to attach ubiquitin molecules to its targets. If I put in what in jargon we would call a catalytically dead Listerine, that does nothing. Does that make sense? One last bit of this is, a detail, but you come across this a lot. You'll see that whenever people overexpress things in biological systems, be it cells in culture or whole animals, you often see things like FLAG or HIS or MYC. Those are things that, through genetic engineering, researchers attach to their protein of interest, creating a handle that or a tag that they can later use for other applications. So for instance, FLAG is a fairly common protein tag that they attach to Listerine that maintains the function of the tag protein, because this is just a short sequence of peptides, not peptides, of amino acids that are attached to the sequence of Listerine. So it's not affecting its job inside of cells, and they can use these tags or handles to later on detect these overexpressed Listerine specifically by looking at the flag, or in some cases use that little flag short peptide as a handle to purify that Listerine from everything else. So not that they do any of that in this paper, but whenever you see flag or 6 times HIS or 6 times MYC, that's what it refers to, these little epitope tags that researchers sometimes attach to these things to work with them later. Questions or comments, anyone? No? Okay. So what these experiments were showing is when you start actually manipulating Listerine, you get a little closer to this idea of causality. And the more Listerine in cells, the less lipids. The less Listerine cells, the more lipids. My question to you guys is, how could this happen? How could Listerine, in a way, affect the levels of lipids within cells? And I'm not asking you about what the authors found. I'm asking you what Listerine could be doing to affect the levels of lipids within cells. Yeah.
I'm pretty sure using the degradation to tag lipids so that they can be uptaken to be degraded.
Okay, so what you're saying is Listerine could be tagging the lipids for degradation. Did I get that right? Yeah? Tagging the lipids for degradation. That could be one mechanism. Jessica? So Listerine could degrade an exporter, right? No, it would lead to more.
Okay.
But the point is, if this is what Listerine is doing, right? So the less Listerine you have, the less degradation. Oh, I see. Yeah, we got, so how about we degrade the inhibitor, or let's degrade the importer. Good point, good catch. Let's degrade the importer. And the argument still stands. Right? Degrade the importer, for instance. Or it could degrade an inhibitor of an exporter. Uptake pathways, right? Well, so in a way, these would be an example of that, right? But yeah, Listerine could affect, alters uptake. Right? Yeah. As you can see, you guys are touching on all possible mechanisms by which you would be affecting, at the end of the day, the amount of lipids present in a cell. So if we think of a cell as this is an analogy that you see a lot, right? Let's imagine that the cell is like a bucket, right? With a spigot at the end, and you have, whatever liquid in it. The amount of liquid present in this bucket will be a function of whether or not the spigot is open and letting liquid through, and how much liquid is being put into this bucket, right? So you could control the amount of liquid present in this bucket, or the amount of lipids present in a cell, by regulating the input of lipids into those cells, the output of lipids from those cells, or some sort of combination of both. Does that make sense? And this intellectual exercise that we just went through is something that the authors must have gone through, and we can tell because of the experiments they tell us they did. They must have thought of a number of possibilities. They did the experiments to address each of those, and they share that with us. So what is that they found? They found that when it comes to uptake, there was no difference between cells that did have Listerine in them, or those cells that were obtained from Listerine mutant mice through the Acrylox trick, right? So what they did is they isolated macrophages from mouse or mice with Listerine or without Listerine, grew those macrophages in culture for a little while, and then they bathed those cells with a fluorescent I'm not quite sure about the details, but it's a fluorescent LDL. And the reason they use LDL, which is a protein, instead of looking at, let's say, cholesterol or other lipids directly, is because in mice and humans, cholesterol is never floating around by itself. It's always bound to either LDL or HDL. That's why if you guys ever go to the doctor and you get some blood work done, you'll see counts of LDL and HDL as sort of surrogates for cholesterol levels in the blood, right? So same thing here, they incubate with some sort of fluorescent molecule attached to LDL, and then they incubate the cells for a little while in this soup with fluorescent, let's say, lipids, I'll call it that. And then after a little while, they wash all of that soup away, and they look inside the cells how much fluorescence was incorporated into the cells, right? So here they show us the images, but more importantly, the quantifications. And the take-home message from this experiment is that after a while of the cells, with or without Listerine, being in the presence of lipids, the amount that they took up inside of them is not significantly different between the two groups. Right? So whether cells have Listerine or not won't affect their ability to take up lipids from the environment. So uptake, not affected. The output, though, seems to be quite different between the two. So for this experiment, they sort of reverse the approach. They let the cells take up all of these marked lipids. They wash away any marked lipids that was present in the culture media. And then instead of looking into the cells right away, they let those cells be for a little while, for 4, 8, 12, all the way through 16 hours. And at each time point, they will take a sample from the culture media and ask the question, how much lipid do I see in this culture media now? Because that's a reflection of how much lipids were being pumped out from the cells, right? So by doing that, they're measuring cholesterol efflux, again, to ApoA1, so cholesterol won't go out of the cells free and on its own. It needs to have some sort of transporter protein on the outside, so the investigators provided that in the form of ApoA1, and now that cholesterol can be handed over to ApoA1, it's being pumped out from cells. And they measure, and in wild types, there is very high levels of cholesterol efflux from the cells, but cells that lacked Listerine consistently show significantly less efflux of cholesterol than wild-type counterparts. Does that make sense? So together, these two observations are saying that Listerine is not important for cholesterol uptake or other lipids, but it seems to be important for effluxing cholesterol from the cells, from macrophages. Questions or comments, anyone? Can I ask everybody to take a look at this graph for a few seconds and tell me if you notice anything about that graph? I know at first I tell you, these data show that there is no efflux of cholesterol. And you're like, oh, yeah, I see it. But now that I'm asking you to more intently look at that graph. Arshia?
In 36 hours, KO is much more than because it's 1.5 versus 1.
Yeah.
One is
Yeah. You guys see it? So basically, Arshia is pointing out that at 36 hours, this thing got really close. Right? And when I saw this data, I'm like, well, how do you explain this? Because a mouse lives longer than 36 hours, right? So had they looked at 48 hours, 72, 96, like 3 or 4 days later, would these two curves have met up with one another, in which case there is no difference? So in my mind, I went into the materials and methods to see if there could be some technical details that might explain this. I didn't find anything. And again, it wasn't addressed in the discussion. But in my mind, this was a bit of an unexplained gap. Like it seems like as she was saying, the KO curve is trending towards the wild type. So will it catch up with it? In which case, after a couple days, it makes no difference having listerine or not.
Last time you were saying that, because this was a flock strain, right? You remember?
Yes.
So last time I remember we said some of it could have still been in there. So maybe those levels started getting higher over time.
Love that. Now, so I absolutely love your argument. I would counter argue saying, well, but the removing listerine with Cree logs happened before we started these measurements. So your argument would be that maybe it wasn't fully removed, right? Which would explain why this curve would have started a little higher. But what I would expect to see is that the slope of the curves are the same. Even though the knockout is lower because most of the cells, but not all, lost listerine, the ones that still have listerine should work as wild types, even though at a lower level. In other words, if your argument is correct, I would expect to see something like this. But what we're seeing is this weird ramping up. Does that make sense? So I don't know. Josh.
Would the accumulation of the lipids within the cell activate some sort of allosteric regulator that activates other proteins that flushes it out?
Excellent. So the comment here is maybe, so the control cells will get rid of that accumulated lipid quickly. Huh? And I need you to sort of go back to this idea that both cells took up the lipid to comparable levels, right? So both cells are equally full of lipids. But the wild type cells are getting rid of the lipid kind of quickly. Within 8 hours, they got rid of a bunch of it. And then they continue effluxing. The listerine knockout cells don't. So they retain those high levels of lipids. And that maybe is triggering some sort of alternative mechanisms for taking cholesterol out. Maybe the cells are like, wait, why is all this cholesterol still hanging around? We should take it out. And then they would turn on other genes or other pathways to try and get rid of it. I like that. I think that from a sort of a biomedical research perspective, the question stands though. Okay, let's say that it's through some other mechanism. At the end of the day, does it even make a difference to target listerine from a therapeutic perspective? Because what this data is showing is that early on, within hours, it does make a difference. But when I do some sort of therapy, I expect the therapy to last for days, right? We usually try not to treat patients every 4 hours. Right? So if it is true that listerine inhibition or deletion, like listerine not being there, works really well early on, well, really well as in it's really keeping the cholesterol in, but then later on some alternative mechanism compensates for that, at the end of the day, it doesn't really make a difference whether an organism has a functional listerine or not, because something else will come and replace it. Right? So, but I like the argument of maybe something is kicking in and doing the job that listerine can't do because it's not there. All right, I just wanted to bring that to your attention because sometimes it's a good habit to really look at the data and see whenever you see something like, wait a second, there is something strange here, that usually triggers more thoughts. So what about the counterpart? We said that the loss of listerine keeps cholesterol inside of cells, at least for a few hours. Will the overexpression of listerine pump out lipids more than in controls? And apparently the answer is yes. Again, they took cells that overexpressed listerine or ubiquitin ligase dead version of Listerine in cells, and compared to controls, the wild-type Listerine pumps out a lot more cholesterol than the ubiquitin ligase dead version. So the data hold in both directions. And then finally, because all of these experiments in culture are nice and very informative, by the way, I should say that I was really impressed with this paper because how thorough they were between going like in vitro to in vivo and back. It's a very it's a very well done paper. So like if you look at it from the perspective of how they follow all the logical steps and did all the controls, it's pretty impressive. So they decide to move into in vivo, and they say, okay, it's fine to see cells pump out cholesterol in a petri dish. Can we see the cells pumping out cholesterol within the context of an animal? So the experiment that they did to check for this is as follows. They took macrophages, and they treated those macrophages with control or scramble siRNA, meaning an siRNA that won't really target anything. It's like one of those sham operations, same thing here. So you treat them with an siRNA, but it's not inhibiting anything, or siRNA against Listerine. In other words, they took macrophages from an animal or macrophages in culture, and in one of them they didn't do anything, and the other one they inhibited the expression of Listerine. And then they filled them up with cholesterol. This time the cholesterol was radioactive. Cholesterol had been synthesized with tritium in it. So you can detect that radioactivity associated with the cholesterol later on. Right? So they fill up the macrophages with this radioactive cholesterol, and they inject the macrophages with radioactive cholesterol into a mouse. And a while later, they basically ask the question, how much radioactivity coming from the tritium can we find in different organs? Why? Because the more cholesterol these macrophages were releasing into the mouse, the more of that radioactivity you would find in different organs. So they went and first checked in blood, then they checked in the liver, then they checked in feces. And in all cases, if the macrophages had been treated with the siRNA against Listerine, there was a lot less radioactivity going around in the mouse. And that's an indication that those macrophages with less Listerine in them were pumping out less cholesterol into the rest of the animal. In this experiment, before they had the cells in culture with these fats or cholesterol in them, and they had a system to measure cholesterol outside of the cells, right? In this case, what they're doing is they're filling up cells with cholesterol, injecting those cells in a mouse, and then they're measuring how much of that cholesterol was pumped out from the cells. And they do that by following the radioactivity that is associated with cholesterol. Does that make sense? So they're essentially observing in vivo the same thing they had seen in vitro. And it's a pretty powerful message that these macrophages with less Listerine in them, these macrophages were not complete knockouts, but they had less Listerine in them, were pumping out less cholesterol than normal macrophages. Cool. So they decide to take it the next step, and they say Listerine is an E3 ubiquitin ligase. It must do what it does by controlling some other proteins, right? We go back to this idea that if we know that this Listerine thing has a catalytic domain for attaching ubiquitin molecules to other things, then it must be doing what it does, facilitating the pumping out of cholesterol by controlling others. And before you had mentioned if Listerine could be ubiquitinating lipids themselves, and you mentioned maybe Listerine ubiquitinates lipids and induces their degradation or something along those lines. Interesting idea, except that ubiquitination is something that happens to proteins. It cannot happen chemically to lipids, right? I just wanted to bring it up because conceptually your idea was fine, but from a technical perspective, you cannot ubiquitinate a lipid. You can only ubiquitinate another protein, okay? So in this case, the thinking is Listerine is affecting the efflux of cholesterol, cannot be working on cholesterol directly. It must be ubiquitinating and therefore controlling some other protein that controls cholesterol in cells. And the obvious candidate was ABCA1. So in this first experiment, they show us a Western blot for a whole bunch of known cholesterol transporters, so ABCA1 being one of them, but then ABCG1, a cousin of A1, SRA1, SRB1. All of those are different proteins that are known to control cholesterol levels in cells. And the samples they worked with are basically cells, macrophages that are Listerine wild type or knockout, right? And that have been stimulated with high levels of lipids in the culture media. In other words, it's like I take these macrophages that have Listerine or not, and I bombard them with lipids, and then I see what happens to the expression of these known cholesterol transporters or lipid transporters. And to make a long story short, the only one protein that changed significantly in abundance as a function of whether or not the cells had Listerine in them was ABCA1. So you'll notice that 6 hours after bombarding the cells with lipids, ABCA1 expression is significantly increased in wild-type macrophages, but if these macrophages did not have Listerine in them, there was no such increase in ABCA1. 12 hours later, same thing. All of the other ones, the levels of these proteins changed one way or another, but there was never a difference between having Listerine or not. So Listerine was controlling the expression of ABCA1 somehow. The first bar graph here is densitometry from the Western blot, and then the second bar graph here is very important. They looked at the levels of ABCA1 mRNA because they see differences in protein, and often we've seen already in this class that whenever you see differences in protein, you're curious to see if those differences in protein may come from differences in mRNA levels, which may be reflective in turn of changes in, let's say, transcription or mRNA abundance. But in this case, it's important for them to show us that the levels of the ABCA1 mRNA are not changing. There are differences in the level of protein that you can find within those cells, but not in the mRNA, right? So that's an indication that whatever Listerine is doing to ABCA1 protein is not being done at the level of the mRNA. Sounds good? And as usual, the converse experiment that was looking at cells that had or not Listerine, they did the experiment in which they put in more Listerine in these cells. And lo and behold, when you put in more Listerine in cells that you incubate with PBS, there is an increase that is significant. So that's shown, that is not significant, sorry, but you can see that there is an increase. So it didn't pass statistical significance, but there is an increase. But when you treat those macrophages that are purposely expressing more Listerine, this increase in ABCA1 is much higher still and becomes significantly larger than in cells that you didn't put in anything in, or cells in which we, as before, put in this catalytically dead Listerine that cannot attach ubiquitin to things. Okay? So you see the gymnastics of this paper is always the same. Let's take out Listerine and let's put in some more Listerine, and what they change is the question they're asking, right? The first question was, what happens to lipids in the cells? Then came the question of, is it influx or efflux of these lipids? And now they're asking questions about ABCA1 expression, but the mechanics of the paper is normally the same. So that RT-qPCR experiment in which they measure the levels of mRNA indicated that mRNA levels weren't changing, but the levels of protein were, so it has to be either translationally or post-translationally. So how, just like I asked you before, an open-ended question, how could the expression of ABCA1 be regulated translationally or post-translationally? Ubiquity degradation is one of the mechanisms that we discuss, right? What other mechanisms for post-translation or translational control can you think of? microRNA, the microRNA, yeah, like MI33, right? But the problem is that that would have oh, you're saying that the mRNA controlling translation, but not the, I see, ooh, I like that. And you even mentioned MIR33, which we saw controls ABCA1 expression a few weeks ago. I like that. MIR33 or X, right? Controlling translation. I like that. And let's go with that for now. Yeah. Protein stability. So how would that be different from the ubiquity degradation? I agree with you, but how can you control protein stability via a mechanism that is not ubiquity degradation? So some sort of post-translational inhibition without degradation. I like that. Yeah. All right. So once again, they must have considered all of these different possibilities, so then they needed to address it experimentally. And the way they do that is by what is called a cyclohexamide chase experiment. The idea goes something like this. If you wanted to look at the rate of degradation of a protein, but you know at the same time that new proteins are constantly being produced at the same time, how can you look inside of cells and say this protein is being degraded faster or more slowly if looking at the levels of those proteins may not be accurate enough, since some of the protein may be destroyed but some new protein is made? So how do you get around that problem? Problem is you'd like to see how fast a protein is being destroyed, but new protein of the same kind is being made all the time. So how can you tell? Label the new ones. Okay. Will that tell you anything about the destruction of the old ones? The destruction of the old ones, the degradation of the old ones, labeling the new ones. Beautiful. So the flip side of that argument, right? So instead of tagging the new ones, let's tag the present ones, right? And then all we need to do is look at the present ones by looking at the tag. It's very, very hard biochemically to get into cells and tag proteins. It's not that easy. The trick is the cyclohexamide chase experiment, and it comes down to, if I know that a given protein is constantly being destroyed but also constantly being produced, and I would like to learn about the rate of destruction, what I need to do is stop any new production, right? If I stop the production of new molecules, then it's almost like the experiment that you were proposing, right? I simply follow the ones that were there already. But we need to stop the production of new proteins. And that's where cyclohexamide comes in. Cyclohexamide is an antibiotic that works by inhibiting translation, right? So the experiment is you take your cells, you incubate them with cyclohexamide. Basically, you stop any production of any new protein because it inhibits translation broadly. And then you simply, with Western blotting, you follow the fate of your protein of interest from there on. So that's why it's called a cyclohexamide chase. You stop the production of new proteins and then you chase the ones that are already there.
And that doesn't affect the proteins that are there?
Absolutely. And that's why cyclohexamide is a poison for cells. But we can use it experimentally to ask this very specific question. So if you take cells and you treat them with cyclohexamide for more than 20 hours, or you expose these, by the way, cyclohexamide, when we say antibiotics, we immediately think of things that kill bacteria, but this is a eukaryotic cyclohexamide. Cyclohexamide is a eukaryotic antibiotic. So if we were to take cyclohexamide would be in the ER within a couple of hours, right? It's really toxic. But in this context, where we're simply asking an experimental question, it doesn't really matter if the cells are dying in front of you. What you're asking is what's happening to Listerine, right? So they did a cyclohexamide chase experiment, and what they saw is that from the moment they gave cyclohexamide, in the wild-type cells, which is the red line here, there was a decrease in the amount of Listerine present in those cells over time, which is totally expected because all proteins suffer some sort of turnover, and within 12 hours, the amount of Listerine within those cells was down to about 30%. The decrease in cells that didn't have Listerine was even faster. And I should have said that these are the levels of ABCA1, by the way. I pointed to Listerine before, but the one that we're interested in is ABCA1. So when you follow the levels of ABCA1 in wild-type cells, it decreases. But if those cells don't have Listerine, it decreases even faster. Okay? So it sort of goes along with this idea that Listerine is controlling the amounts of ABCA1 within these cells post-translationally by, and what would you say? What is that Listerine is doing to ABCA1? Is it marking it for destruction? No? You have your hand up? No? So broadly speaking, is Listerine normally protecting ABCA1 or is it hurting it? Protecting it, right? And we see that from the fact that we took out Listerine and now ABCA1 was a little more defenseless. It was being degraded faster than in the presence of Listerine. Yeah? All right, we'll leave it at that. There is only a few slides left, but we'll do it on Wednesday as usual. For those of you who didn't get a chance to ask or answer a question, the assignment will be available soon. It relates to one of the things that we discussed in class. And if you have any questions, as usual, I'm going to hang around for a little while. If not, I'll see you Wednesday.